our speaker, Melissa Emery Thompson, who's in the Department of Anthropology at University of New Mexico. She's also the chair and co-director of the Comparative Human and Primate Physiology Center there. And as many of you know, she specializes in grade 8 behavior and biology and examines the interactions of ecology, health, reproduction, and behavior in both field and lab settings. And it's really great to have her presenting here at Club EvMed. And our discussant for after Melissa's talk is Arun Karlamangla. He's a professor of medicine at UCLA, and he's board certified in both internal medicine and geriatric medicine. He conducts research on physical and cognitive decline in older adults. He also looks at the determinants of successful aging and the role of psychosocial stressors in the health of older adults. And so it's a pleasure to have you both here today. I'm going to turn things over first to Melissa, and then we'll hear from Arun. So go ahead, Melissa.
Great, thank you. Hopefully you'll let me know if my audio or anything about the slides looks funny. And am I sharing the right screen now?
Yeah, great.
Okay, excellent. Thank you, Charlie. Thank you, Meredith. It's nice to be here. As Charlie said, I'm an anthropologist. I'm not a clinician. So this research direction into health and aging in chimpanzees is fairly new for me. We've been working on this project for about 6 years. And so I'm really excited to speak to a different audience and kind of get a sense of where this research could go and what other disciplines are interested in about it. So we've just entered what the WHO is calling the decade of healthy aging. That's accompanied by all sorts of really hilarious stock images like this one, but public health organizations worldwide are really focusing on the rapidly growing population of older folks. And this concept of healthy or successful aging is really kind of a philosophical shift away from aging research that was sort of focused on prevention of aging towards this idea that maybe we can think about what leads to improving the lives of older folks and increasing the number of healthy years. So I want to focus here on what chimpanzees could tell us about healthy aging. So animal models have long been used in aging research, but they're overwhelmingly these small, short-lived organisms, things that are chosen out of convenience often because they can be manipulated easily in the lab and have short generation times. There's also been an emphasis on species that have peculiar anti-aging mechanisms. And so we've learned a lot from these models about mechanisms for aging broadly in biology. But there are serious limitations of many of these models on their applicability to humans. So, while hunting for translational applications from other animal models is important, I think that animal models can also really help us understand just the basic biology of aging and how it evolved. And so compared to other model organisms like mice or flatworms, species like primates really offer a higher likelihood that the physiological mechanisms of aging are shared with humans. And since humans are primates, these most closely related species will actually share these mechanisms because of a shared evolutionary history. So in other words, it's more likely that mechanisms that are apparently similar really do work in the same way and for the same reasons. Also, this shared ancestry and all these similarities make an even more powerful context in which to look at the differences between human aging and those closely related species. So these might indicate, recent selection, for example, that accompanied the extension of human lifespan, or they could indicate critical changes in context, like differences in the way that humans produce and use food. Primates are obviously also essential models for aging because they're long-lived compared to comparably sized mammals, and because they live in complex social systems. So we can study attributes like social hierarchies and social support that are known to profoundly affect aging in humans. So this is just an example here. This comes from our recent special issue on primate aging. The study by Guevara examined similarities in age-associated epigenetic change in chimpanzees and humans. This figure on the left here shows you the extensive overlap in the age-associated methylation with age. This is overlap in the sites that become methylated. And the vast majority of these sites age faster in chimpanzees than in humans, which of course makes sense since this is a shorter-lived species. But the genome-wide methylation levels have been used to predict people's ages with remarkable precision. And it turns out that if you adjust for differences in lifespan, the clocks that have been used from humans can actually predict chimpanzee ages as well to within 3 to 5 years. And that's what you see here. So this points to really extensive similarities in the physiological aging process. It's just another example of a recent study that looked at physiological dysregulation, which is kind of an integrated index of changes in homeostatic regulation associated with aging. And what you're seeing here is the strength of the correlation between results when a species is used as a reference for itself versus when it's used as a reference for another primate species. And the area in the box shows that how each species would perform as a reference for humans. And you can see that even across primates, the species that are most closely related to humans, especially chimpanzees, make more appropriate references for evaluating physiological change with age. So those studies necessarily involved captive chimpanzees who live very different kinds of lives than their wild counterparts. So a second important feature of an evolutionarily relevant model is to replicate as close as we can the kinds of environments in which the aging process evolved. And we've seen that concept used to great effect in research that's coming out of populations like the Chimani and other small-scale societies, where anthropologists have been doing research on aging in diverse ecological settings. So the Chimani are a horticulturalist population in Bolivia. They have very little access to medical care, but they show almost no evidence of coronary artery disease. So something that we maybe had accepted as an inevitable risk of aging appears not to be, after all. And so I think medical researchers could probably have predicted or already knew that lifestyle factors affect the risk of cardiovascular disease, but it was kind of a revelation that things like healthy diet or routine physical activity might actually prevent it altogether. And now there are a number of other studies of small-scale societies that have been piling up to indicate that a lot of the degenerative diseases of aging may have increased only in our recent history or in industrialized settings. And so these small-scale societies often suffer poor life expectancy, which is usually due to infectious disease during childhood. But the older folks in these societies are in remarkably robust health. But we still have very little data on aging in the species that are most closely related to us, the great apes. The life course of these species is greatly extended compared to other primates. So all of the great apes can live past the age of 40 in the wild, and for chimpanzees, individuals can live well into their 60s. So there's a lot of reasons to expect that selection for longer lifespan in these great apes might have entailed significant changes to aging biology, and that this is pertinent to understanding the evolution of human aging and is not represented in common laboratory animal models. So with that in mind, for the past 6 years, I've been leading a collaborative team, you can see a pretty large team, on an NIA-funded study that's really the first of its kind to examine the biology of aging in wild and free-ranging chimpanzees. So I want to thank everyone up front because the work that I present today is a lot of their effort. So this project has been taking place in 2 different settings. The first is in the wild. This is the major component, and occurs in the Kibale National Park in southwestern Uganda. Here we study 2 wild populations of chimpanzees, sorry, two wild communities of chimpanzees that number over 200 individuals. These animals are completely wild, so we don't feed them, we don't manipulate their environment or their behavior in any way. Even things like medical interventions are pretty rare and reserved only for treating traumatic injuries. The advantage of the wild setting is we can monitor the natural behavior, the natural activity of the chimpanzees and look at all the challenges that they would normally face. But on the other hand, we're constrained by doing things that are completely non-invasive. So our work consists of daily observations of the chimpanzees and sampling for urine and for feces. And that being said, these samples of urine and feces can actually yield a pretty robust set of measures that are relevant to the biology of aging. Now, this particular study started in 2015, but these chimpanzees have been monitored for 20 or 30 years. So we actually have this really extensive record of sociodemographic data and even physiological data that rivals, I think, even some of the best human cohort studies. We also do work in African chimpanzee sanctuaries. These are like rehabilitation centers that take in young chimpanzees that have been confiscated from the pet trade or the bushmeat trade. And it essentially provides them a safe place to live out the rest of their lives. So all the chimpanzees, nearly all of the chimpanzees in these sanctuaries were born in the wild. And they have secure indoor sleeping enclosures, but otherwise they have large naturally forested areas where they spend their day in social groups. These are usually large islands. So the chimpanzees are provisioned, but their diet is much closer to what a natural chimpanzee diet would be than than the chimpanzees living in zoos or labs. They eat lots of fruit and vegetables, and they're not fed these kinds of like high calorie monkey chows that are used in zoos. So these aren't wild chimpanzees, but they live as similarly as possible. And as part of the management of these sanctuaries, they conduct veterinary exams under anesthesia about once a year. So our researchers took advantage of this to go collect blood and other kinds of samples and to do point of care testing that wouldn't be possible in the wild. So the first study I want to tell you about comes from this setting and was done by my PhD student, Megan Cole, and Alex Rosati and colleagues. They participated in these health checks and examined the blood lipid profiles of the sanctuary chimps and then compared them to publicly available data on these markers in US laboratory chimps from places like Yerkes that are stored in the NIH's primate aging database. So here are the data for total cholesterol, LDL cholesterol, and triglycerides. The dashed line on each graph indicates the level that would be considered risky for human cardiovascular health. The captive chimpanzees, which are in blue, frequently met or exceeded those risk thresholds, while the sanctuary chimps, which are here in green, rarely did. They were well under it, so only one individual seems to cross that risk boundary. So the older chimpanzees also, as they age, got fatter, the captive chimpanzees, as they age, got fatter and exhibited riskier profiles, but that wasn't the case in the sanctuary chimpanzees. So for a long time, it was assumed that there was actually a species difference in these blood lipids, such that chimpanzees just naturally had higher cholesterol than humans. But these new data suggest that that's probably not the case. And so captive chimpanzees have blood lipid profiles that look like humans in sedentary societies like the US. And here you see in yellow the some comparisons. But the sanctuary chimpanzees, whose lifestyles are closer to what they would be in the wild, have levels that are similar to humans in many small scale societies. So these data really reinforce that lifestyle factors could act in the same way in chimpanzees and humans to affect this underlying risk for cardiovascular disease. And it adds to data from the Chimani and the Hadza that cardiovascular risk doesn't necessarily need to increase with age. Now, an important point to make here is that chimpanzees actually don't appear to be susceptible to ischemic heart disease. They don't get clogged arteries. They do die of heart disease in captivity, but it's typically myocardial fibrosis. So it doesn't seem to be related to age, and it's not related to these blood lipid levels. And this difference has gotten a lot of recent attention because it suggests that the human heart has undergone some recent evolution that makes us more vulnerable to this particular kind of coronary artery disease. So here we have an example of where we seem to have inherited the underlying physiology that promotes risk from our ape ancestors, but it's this unique combination of recent evolution on the heart and sedentary conditions that is leading to what is now one of the most common and deadly diseases of aging. So we can't measure these blood lipid profiles in wild chimpanzees, but in the past couple of years, we've really focused on trying to examine this constellation of interrelated features that are associated with risk of age-related disease in humans. And these features are commonly linked as characteristics of the inflammaging pathology in humans, and each factor kind of exacerbates the others. We know now that immunosenescence is a clear feature of aging in wild chimpanzees. They have, as they age, they have higher viral and parasite loads and increased incidence of clinical signs like coughing and sneezing. This isn't really particularly surprising because immunosenescence seems to be a broad feature of aging across all sorts of species. Another thing that we see in common with humans is evidence of increased HPA axis dysregulation with age. This is a characteristic in humans that's thought to really exacerbate the negative effects of inflammation and to promote all kinds of degenerative diseases. So here's a recent paper where we examined a data set of over 15,000 urine samples that we'd collected from the chimpanzees over 21 years of study. So we could not only track these longitudinal changes in individuals, but we were able to control for socio-demographic factors like reproductive condition or social status. And we found robust increases in cortisol in urine with age in both sexes. And actually, of all the things we've looked at over years of looking at this, age seems to be one of the strongest influences on glucocorticoid levels. But more importantly, on the right here, we found significant age-related changes in the diurnal rhythm of cortisol production. This is strongly indicative of dysregulation as opposed to just having more stress as you get older. And the blunting of the diurnal rhythm that you see here looks very similar to what has been observed in humans and suggests a common origin of that process. Despite this, so we have this associated risk factor for inflammaging that seems to be present in chimpanzees. We found no evidence that wild chimpanzees suffer from age-associated increases in either inflammation or oxidative stress. And we've looked at multiple markers of these things. These markers will increase temporarily when chimps are ill or injured, and they do increase in the 2 or 3 years before older chimpanzees die. So inflammation is important, but what we don't see is this evidence of this chronic low-grade accumulation of inflammation that is what seems to yield health risks in humans. Finally, a big focus of research on successful aging has been focused on the frailty syndrome, which is kind of a suite of characteristics of declining physical condition and physical performance that are associated with disabilities in performing what's called the activities of daily living, but is also a robust predictor of morbidity and mortality in aging human populations. So we can't perfectly replicate the diagnostics for frailty in the wild, but we can assess changes in physical activity, and we can also look at physical condition using urinary creatinine, which is a well-validated measure of muscle mass. And again, we can apply many years of longitudinal data on the same individuals to answer this question. And so we do see declining muscle mass with age in both males and females, but these changes are actually relatively moderate until the mid-40s or so. We also see some age-related changes in physical activity, but again, they're pretty moderate. So older chimpanzees spend about 30 to 40 minutes less time in the trees over the course of a day, which suggests they could be reluctant to climb or have difficulty climbing. They also spend about 15 minutes less time feeding per day on their preferred food, which is ripe fruit. But even at the oldest ages, chimpanzees are spending well over 3 hours a day eating fruit. So 15 minutes is not a big difference. Age didn't affect the amount of time that the chimpanzees spent walking, nor the amount of time that they spent resting. Now, in humans, the important feature of frailty isn't that physical condition declines with age, but it's that it has a disabling effect on individuals. So what we did was examine whether those chimpanzees whose muscle mass fell below a threshold of 1.5 standard deviations from the youthful average, whether they experienced negative effects. And the results were overall pretty weak. We found that poor muscle mass didn't affect most kinds of activities like walking, feeding, or climbing. Older individuals with poor muscle mass did rest a bit more, but it amounted to about 6 minutes more per day. That's a little nap. As a proxy for morbidity, we looked at respiratory signs like coughing, which are the most commonly observed clinical indicator in our population. And we found that males who had poor muscle mass did experience more respiratory signs, but the data for female actually were in the opposite direction. The smaller females had fewer signs. But most importantly, any way we tried to look at the data, poor physical condition didn't predict mortality as it seems to in human populations. So if we take all this together, these data are pretty striking because chimpanzees live in really physically demanding lifestyles. They experience high loads of parasites and other sources of infectious disease. And they don't do what humans do, which is to care for each other when they're old or infirm. And so we found that chimpanzees experience the effects of aging as an increase in the burden of infection and in challenges to the systems that maintain homeostasis. But they don't appear to suffer from chronic inflammation or from accumulated frailty. So we have sort of the ability to pull apart these interrelated features of aging. The last thing I want to talk about is a recent study we published that deals with social aging. Nearly every program that targets successful aging emphasizes this problem of social isolation in the elderly. And this is kind of a vicious circle because a lack of social connections has been shown to have pretty profound negative effects on health. And yet most people, as they age, at least in industrialized societies, become progressively socially isolated. Now, one of the prominent theories that tries to explain this phenomenon comes from clinical psychology, and it's socio-emotional selectivity theory. And the idea here is that as our concept of the future shrinks, we shift our motivations to focus on the social interactions that provide us with the most positive rewards. And so a central premise of this theory is that it's not aging per se that leads us to narrow our social circles, but it's our perception of how much time we have left and whether it's really worthwhile investing in new relationships or risky social interactions. So this theory necessarily involves these complex cognitive processes that are unique to humans. So humans exhibit three key features of aging. The first is that we have shrinking social networks, meaning fewer connections. Second is that we tend to invest more heavily as we age in those most important and most emotionally rewarding relationships. And finally, we exhibit a strong positivity bias, which means that we selectively attune to positive versus negative social stimuli and engage in positive types of interactions over negative ones. So there's evidence now from all sorts of primates that suggests that social networks commonly shrink with age, and that older individuals in particular engage less with others in social behavior. But social aging in monkeys does not seem to have the same set of characteristics that are observed in humans. For example, monkeys tend to have an increased negativity bias, characterized by higher use of aggressive behaviors versus affiliative behaviors like grooming. One paper describes older monkeys directing threats at any animals that came nearby, so limiting their social circles in that way. The available studies also make it unclear whether monkeys are prioritizing important relationships or whether they simply have fewer social contacts as they age. So in macaques, for example, a declining number of social partners might simply be a result of the fact that fewer and fewer of your relatives are still alive as you age. And finally, a cognitive study of Barbary macaques indicated that aging individuals were still very interested in social stimuli, much more so than environmental stimuli, which suggests that the changes in sociality could be more of a product of constraint. In other words, they were becoming socially isolated rather than being selective by active preference. So we again applied our long-term data set to address social aging in chimpanzees. Here we focus on males because in chimpanzees, males have more well-developed social connections than females do. We found that older chimpanzees had fewer social partners, like monkeys and like humans, and were more likely to be found alone than in groups. But interestingly, when they joined groups, they were more likely to be in close proximity to another male. And this was true regardless of their social rank. And so these different blue boxes on the figure show the different levels of the social hierarchy. So this active engagement with social behavior is already different than what we see in many primates. Additionally, older males had a higher number of mutual friendships and fewer one-sided or unidirectional friendships as they aged. And so what this means is that two individuals showed a strong mutual preference to associate with one another as opposed to other individuals. And we found that these relationships were characterized by not only higher rates of social grooming, but an increased equitability of the grooming that you gave versus the grooming that you received. And older chimpanzees not only formed these mutual friendships with other old guys, but also with young individuals. This pattern is much more indicative of selectivity and prioritization of important relationships than with social isolation and what's been documented in the monkey species that have been looked at. Finally, in contrast to the monkeys and more similar to humans, chimpanzees showed a positivity bias. So they engaged in less aggression as they aged and had more positive social interactions. And it's worth noting that all of these patterns are independent of the male's social status. So that tends to decline in later years, and yet they maintain positive social interactions. So taken together, these data suggest that social aging occurs broadly in our close evolutionary relatives, even though they lack a well-developed sense of the future or of their impending mortality. So this means we have to sort of re-examine these existing theories to consider the evolutionary origins of social aging and how it might relate to senescence more generally. These changes in humans may be guided by complex cognition and by emotionality, but they're likely layered on top of patterns that have evolved in response to the fitness costs and benefits of social behavior that can be expected to change as we age. Now, the pattern in chimpanzees look much more human-like than what we saw in monkeys, and is consistent with active selectivity. And one reason we might see this different pattern, I think, is that in most primates, you get these cohesive social groups that stay together all the time, and that makes it a lot more difficult to choose to engage in rewarding interactions while also avoiding those risky interactions. And so this increased negativity and the withdrawal from the social center of a group could be a coping mechanism just to avoid risk. But chimpanzees are a bit more like humans in that their social organization is fluid. So they can choose to associate with particular individuals, and they have a lot of flexibility to decide who they want to associate with and when. So chimpanzees experience social aging, but they don't appear to be isolated. They appear to be maintaining healthy, rewarding social lives late into life. Finally, I think it's worth considering that social isolation as kind of a pathology of the elderly could be like a lot of the other diseases that are discussed in evolutionary medicine, where this could be a result of mismatch, where we have strategies that evolved in the context of successful aging in the past that are poorly suited to the recent changes in social environments in the developed world. So what I tried to do today was to provide some highlights of the ways that evolutionarily relevant models can improve our understanding of human aging. So this is a very different approach to the use of animal models. We're probably not going to learn how to cure aging through this kind of research, but I think it helps us understand how other long-lived organisms deal with the challenges of aging, and clearly can help us try to disentangle the natural processes of aging from the things that are novel or pathological or highly sensitive to lifestyle changes. So in this case, we've been able to examine 3 of the most prominent paradigms in research on successful aging: inflammation and cardiovascular disease, frailty, and social isolation, and show that our closest living relatives living in the environments in which they evolved may not live as long as humans, but they age successfully by human standards. So our emphasis in the first phase of the study has really been to document how chimpanzees age, and as we move forward, we're going to be leveraging the really detailed longitudinal nature of our data set to try to understand how different life experiences for the chimpanzees lead to variation in health. So, if they're not dying of heart disease and other degenerative diseases that we've come to associate with aging, what are they dying of, and what predicts the length of healthy life in these individuals? And also, now that we have all these baseline data sets, we're interested in more direct comparisons with studies of humans, and really kind of open to people's ideas about what could be done in that domain. So thank you to you for your attention, and I want to acknowledge in particular the long-term field staffs and the sanctuary staffs that collect data on the chimpanzees every day. Thank you.
Okay, thank you, Melissa. That was fantastic. Very nice overview of a lot of different topics. And I want to turn now to our clinical discussant, Arun Karlamangla, who is here with us from UCLA. So Arun, if you're still on, please let us know. You can unmute yourself.
Thank you. Thank you, Melissa. That was fascinating. I was paying close attention to lots of things. From a clinician's perspective, we tend to focus on things that are potentially modifiable, and some, but not all of the factors that you mentioned that relate to how wild chimpanzees age are potentially modifiable and possibly applicable in humans as well. So let me focus on a couple of things that I thought we could draw parallels to. And we'll start with the HPA axis dysregulation, because that's one of the things that my research is focused on. So let's see if I can share my screen. Hold on. I think I can. Okay, you're able to see my screen now?
Yep.
Yeah, perfect. All right. So, Melissa mentioned the diurnal cortisol rhythm, for instance, and that is something that we've studied quite a bit in humans. And so this is a schematic of the diurnal cortisol rhythm in humans. Cortisol levels start off high when you wake up. They peak about 30 minutes after you wake up, and then fall gradually over the day. The picture you see here is something that we used when we fit a linear spline model to model the trajectory of cortisol over the day. And we looked at two particular characteristics. One that we call the diurnal dynamic range, which is the dispersion between the peak and the trough, the nadir, and then the area under the curve, which is the total amount of exposure to cortisol over the day. And what was fascinating was we did not find what people expected, that there is so the blunting of the cortisol rhythm that Melissa talked about. We do see that, but we don't see increased blunting as humans age. In this study, which was from the Midlife in the United States study, which though it's called the Midlife study, had a large age range of people. Looking cross-sectionally, at least, we did not see the blunting of the cortisol rhythm, which means that the dynamic range that you see in this picture did not go down as people aged. However, the area under the curve increased. So there was kind of a lifting of the cortisol rhythm over the day as people aged, which was a greater nadir and a greater peak. On the other hand, getting to what might be modifiable, is lifetime adversity. And here we looked at adversity during childhood, measured both in terms of socioeconomic conditions and abuse, either by parents or friends in childhood. And we created a scale of childhood adversity. And what we see here is that the dynamic range, which is shown by bars, gets smaller and smaller in comparing people's childhood adversity. On the other hand, area under the curve did not increase, so it did not change statistically significantly as a function of childhood adversity. So the blunting of the cortisol rhythm, at least in humans, seems not so much a function of age as much a function of stresses and life stresses. Here we show childhood adversity, but we also see similar things in relation to level of education, socioeconomic status in adulthood, etc. So a little bit of a difference, but maybe suggesting that the blunting of the cortisol rhythm may not just be a natural aging process, but a result of adversity, which may be malleable, which may be modifiable. Now, so why does this matter? So in animal models, and this are rodent models, they've shown that having a nice, robust cortisol peak in the morning is important for hippocampal neurons, dendritic spines in hippocampal neurons, with reflecting learning processes. So to learn new activities, learn new skills, etc., having a robust cortisol peak in the morning seems to be important. Having a low nadir at night is important to retain those memories, suggesting that both a high peak and a low nadir is important. And so if there is a blunting of this cortisol rhythm, that might mean less ability to learn new things, which is one of the things that we see as people age. that when you think in terms of cognitive aging, one of the things to go down rather quickly is memory and making new memories and so on. I'm going to see if I can show you something else here. Hold on. So, yeah, hopefully I'll get better at this. Let's see. How do I stop sharing?
There should be a red kind of box. Yes.
Got it. Okay. All right. And let me try and share something else here. So now in terms of cognitive aging, so this is a different study that we were involved in called the Study of Women's Health Across the Nation, which followed women longitudinally through the menopause transition. And we were looking for early changes in cognitive functioning in these women. And so what we found was that one of the first things to go. We did see changes in memory as well, but one of the first things to decline was cognitive processing speed. And what I wanted to show here was that even as early as age 54, you begin to see declines in cognitive processing speed, and with a little bit of acceleration around age 61. So we see similar things with memory. We see changes starting around age 58, and changes in working memory around age 61 as well. So the difference between these curves here is we were trying to look again at modifiable factors, the associations of modifiable factors with the rates of decline in cognitive processing speed. And what you notice here is that we looked at physical activity because there's been a lot of talk about physical activity helping improve cognitive aging. And unfortunately, we did not see much in terms of physical activity. I don't know if you can see my cursor move here, but these are the changes, the magnitude of the changes in cognitive aging. The top three lines refer to different levels of physical activity. On the other hand, financial hardship was a major player. Women who reported more financial hardship seemed to decline longitudinally in processing speed at a faster rate. The dark brown line here, the dark brown dotted line refers to people who had the highest levels of financial hardship. So again, looking at modifiable factors, cognitive aging seems to be a universal phenomena, but different people seem to age at different rates, and the rates might be related to modifiable factors. If I have a few more minutes, I'm just going to talk about something that Melissa did not see in chimpanzees, but we do see in humans, which is the importance of muscle mass. Let's see if I can find that here. Okay, so where is the sound? Okay, there we go. Okay, so as people age, muscle mass tends to fall. Again, it's not inevitable, but it's the common pattern. And we looked here at whether muscle mass is a predictor of longevity. And we did find that after adjusting, and this is data from the National Health and Nutrition Examination Survey longitudinal mortality follow-up. And we looked at quartiles of muscle mass index. So as people get older, body mass index or BMI is not a great predictor of outcomes in older folks, though it's a major predictor in midlife. It's not so good in older folks. However, muscle mass index is. So what is muscle mass index? It's the total muscle mass, and hence it was measured using bioelectrical impedance. The total muscle mass in the person divided by the square of height. So analogous to body mass index, which is the total body mass divided by the square of the height, muscle mass index is total muscle mass divided by the square of height. And we look at quartiles of muscle mass index in older adults as a predictor of longevity. And we find that the people in the two highest quartiles of muscle mass index have longer survival. So these are the relative odds and relative hazard for all-cause mortality as a function of the quartiles of muscle mass index. So here again, the message from a clinician's perspective is try and preserve your muscle mass as you get older, continue to stay physically active. And it's not enough to just be doing cardio exercises, muscle building exercises, muscle preserving exercises are important as well. So I'm going to stop there and open up for questions.
Very interesting. Thank you. Thank you for sharing those results. Really interesting. The cortisol one that you presented at first, I found especially interesting. I wanted to, first of all, ask anyone who has questions to please raise your hand and I'll call on you. And I just wanted to see if Melissa had any thoughts about the cortisol results or any other results that Arun just presented.
I think that there are a number of studies showing age-related declines in cortisol, but I think that there's a lot of reasons to ask where that comes from. Is it actually age, or is it are there other things going on? And so the idea of heterogeneity in aging of these systems because of things that happen way back when, I think is fundamental. I know that Susan Alberts and her crew are doing some pretty great work on baboons looking at that. We're not quite at that stage yet, because even after 30 years, that's just a half of a chimpanzee lifetime, but it's definitely cool stuff.
Good point. So I had a question just to get things started. We have probably about 10 minutes that we can devote to discussion here. And I'll just start it with a question. You talked about physiological aging, physical aging, behavioral aging. I didn't hear much about reproductive aging. And I wonder if you could say anything about that. And this mirrors some questions that were in the chat. Peter Gray asked about sexual behavior and aging in relation to that. And Joe Graves asked about testosterone. I wonder if you could give any general comments about reproductive aging in males, and Pusey brought up, I'd like to hear about females. So I can bring up all three.
Those are a couple different questions. Peter and I have talked about this before. Older chimpanzees don't seem to have problems with erectile dysfunction. We haven't studied it systematically, but it's certainly not something that seems to be an issue. We have looked at reproductive aging in terms of fertility for both males and females. There's a recent paper that Martin Muller led on age-specific fertility in males. We find, of course, declines with age. So in chimpanzees, or in female chimpanzees, the age profile of female fertility looks pretty darn similar to humans late in life, except that they tend to die right when menopause would strike them. In males, we see huge peaks in early adulthood and certainly a decline late in life, but we have some old guys that manage to sire offspring, and sometimes they sire their first offspring when they're old. But getting at reproductive function as opposed to changes in social status and alliances and all the other things that affect fertility in those older males is tough. We are in the process of doing the testosterone analysis. We've got people in the lab right now working on that, and we're gonna have a data set that is tens of thousands of samples in which to really look at the issue of testosterone with age. I mean, certainly it declines, but how similar that looks to humans, I'll hold off on that. As for the human question, I suspect Anne was asking about the social aging, and we do have a new analysis where we're looking at social network measures for females. Really, the gist of it is when they're old, they at least in our population, they have almost no social connections. And so what few that they have when they're younger, they seem to just uniformly decline when they're older. And this is weird because, like chimpanzees at other places, females gain in status as they age. So you would expect that they would be sort of more valuable social partners, or have a safer time engaging in social behavior. So they seem to choose not to, and we're still trying to understand what's going on there. But for them, it's almost as if having higher social status substitutes for having strong social relationships.
Is that what you were asking about, Anne, in the chat?
Yes, I think that's very interesting. I mean, I think we'll find the same with the Gombe chimps. So yeah, females just are not very social. Unless they have family members. How about, with their sons and their, I suppose mostly with sons in your community.
Right.
Okay, thanks for that. I had another question as well. You brought up earlier in the talk something about the evolution of the human heart and something that's unique about the human heart. And I think Joe Graves was also mentioning this in relation to some of the work, I think it was on the triglycerides or something, about being a dietary issue more than a change in the human heart. I just wasn't entirely clear what that was about with the human heart and what the evidence is. If you could give any more details on that.
Oh, I'm going to flub all the specific anatomical details. I can't answer that quite precisely. There's good work by Varkey and then a recent paper by Daniel Shave and Dan Lieberman's group that have looked at features of the heart, which suggests that there is really an actual difference in the anatomy of the heart that makes it more vulnerable to this kind of disease. But again, I'm not a clinician, so I don't want to comment too much on that. But what Dr. Graves says about lifestyle is certainly a huge part of it. But again, we have these captive chimpanzees that have terrible cholesterol levels and high triglyceride levels and sedentary lifestyles and crappy processed diets, and they don't die of coronary artery disease. So there's more to it than just that. I think it's a combination of things. So I think Lieberman has argued that this is more about evolution on the heart's ability to sustain endurance activities that chimpanzees, for example, aren't doing.
If I could only quickly get in a comment on that, it's just really hard, it seems, from what I know about the difference between human and chimpanzee hearts to jump to that conclusion, especially since, for all the points that you've made throughout your talk, the environmental experiences of the chimpanzees you've looked at in the field and even under captive conditions in no way, simulate the way human beings live in our society. So, I mean, maybe it's there, but I'd really like to see much stronger evidence of that.
Fair enough.
Great, thank you for that. So it looks like we have a question from Mandy Azar in the chat that I believe is for Arun. Mandy asked, what were the modifiable risk factors you mentioned for the cognitive processing speed you saw in females?
So we investigated a variety of them, including physical activity and financial hardship, and I showed you that the associations with financial hardship, which hopefully is malleable at a societal level, may not be at an individual level. The associations were strong for financial hardship, not so strong for physical activity, which is something that would be modifiable at an individual level. So yes, those are the two that I showed, and they were, and of course, yes, there is good data on smoking being bad. We didn't have enough variability in alcohol consumption in this cohort, so we didn't see an alcohol effect.
Okay, great. I think There's time for one last question. I see it's getting answered in the chat already. Jillian Bentley asked, do captive chimps get any cancers that we know of? Melissa, do you want to answer that to everyone, or?
I mean, what I've seen is just from, there's a few papers that sort of aggregate, necropsy results from captive facilities, and certainly there are cancers, but the data suggests that rates of cancer in chimpanzees are lower than in humans. And I'm not really aware of anything from, now keep in mind that when wild chimpanzees die, our ability to do necropsies is pretty limited. So there are very few, but cancers don't seem to pop up as something that is common in wild chimpanzees. So we should have seen it by now, I guess is my point. So that's another big difference.
Okay, thanks for that. Thank you both very much, Arun and Melissa, for really nice perspective on aging in chimpanzees, aging in humans, how it's relevant for understanding human health, Arun, and medicine. We're at the top of the hour, so we have to wrap up. Thank you all very much for attending today. It's great to see so many of you here. Always fun to have these conversations. And before we go, I want to share with you our next one. Hopefully you're seeing my screen now. I'm sharing the screen from Club EvMed. You can go there and click on upcoming and see the upcoming events. Here's the one we just heard today. And our next one is with Christine Stabile-Ben. She's a professor in global health from the University of Southern Denmark, and she's going to talk about how vaccines train the immune system in ways no one expected. I think it's a fascinating topic, and I hope you'll join us in a few weeks to hear about this talk. And then it goes on from there. We don't have all the descriptions up yet, but those will be coming soon with a great variety of talks coming up, and even a few more that'll probably end up in there. So thank you all again very much for joining us, and we hope to see you at the next Club EvMed and other ones in the future.